Hello everyone, welcome to our lecture today on the functional units of the nervous system, which is just a fancy way of saying neurons. We're going to be talking today about the structure and function of neurons. We're going to start off reviewing things that hopefully have already been covered in your prerequisites but are important for understanding neural function. We'll talk about the basic structure of neurons, how we can understand or classify different kinds of neurons, and we'll also talk briefly about glia, which are the support neurons in the central nervous system, or I'm sorry, the support cells of the central nervous system. All right, so this is a very brief review of cellular physiology. Of course, neurons are cells, just like every other cell of the brain, so you should have a good basic idea of how cells function. If not, please review this material in the textbook or from your prerequisite classes. Probably the most important part of the neuron for our purposes is going to be its membrane. Now, you remember the membrane is made up of this bilayer of these phospholipid molecules. The phospholipid molecules have this polar or hydrophobic water-compatible head with these hydrophobic or water-incompatible tails. And when they're arranged in the bilayer, the hydrophilic heads are arranged either to be facing the extracellular fluid, the aqueous environment, the watery environment of the extracellular fluid, or to be facing the watery environment of the intracellular fluid, and the hydrophobic tails are aligned or up next to one another. Now, the important thing that the membrane does, of course, is separate the intracellular and extracellular space, but a key thing to remember about it is that it is semi-permeable. So what do we mean by that? Well, if you just take the phospholipid bilayer, and in particular the presence of these hydrophobic tails, that makes it a pretty effective barrier against water, against ions, or for any large molecules, or anything else that might want to pass through. So so the lipid bilayer serves as a barrier for most things. But embedded within the bilayer are proteins and all kinds of other things, and for our purposes, they're embedded with channels and pumps that will selectively allow certain ions and other molecules through. And as we'll see later on, it's this ability to selectively pass certain ions and molecules that makes the membrane important for the resting potential and for the action potential, which are sort of the two most important electrical activities that a neuron carries out. But we're not getting there yet, but just to set this up, we're going to be talking about in the future a lot about the membranes, a lot about the cell membrane and its property of being semi-permeable. Finally, you should remember a little bit about how ions work. So you'll know, of course, that atoms usually have a neutral charge because they have the same number of protons in their nucleus as they do electrons in their orbitals. But occasionally, atoms can gain or lose electrons, and when they do that, they acquire an electrical charge, and we call them ions. Two ions that we'll be talking about, for example, are the sodium ion and the chloride ion, which are, of course, the components in table salt. Now, if we look at the typical water molecule here, a water molecule is not an ion. It doesn't have a net positive or negative charge. However, it is polar. Polar means that there are certain parts of the molecule which have a slightly greater accumulation of positive charge, and another region of the molecule that has a slightly greater accumulation of negative charge. This polar property of water allows it to very easily form hydrogen bonds with and to dissolve ions such as sodium ions and chloride ions. And so, of course, as you know from your experience, if you put table salt into water, it will dissolve. And what's happening there is that the polar water molecules are sort of being attracted to and are sort of inserting themselves in between the crystal structure of the salt crystal, is pulling those ions out of the crystal structure into solution, at which point they are going to begin to diffuse away until there is an even distribution of the sodium ions and chloride ions throughout the water. This process of diffusion, of ionic diffusion through aqueous solution, is going to be one of the key mechanisms, or one of the key things that helps drive the resting potential and action potential. All right, so that was our brief review. Hopefully it wasn't too new for you. What we're going to do next is talk about the basic structure of neurons. And just to do that, I'm just going to, if you'll indulge me, do a little bit of history. So what I want you to do is I want you to imagine that you're a scientist living in the late 1800s, and you know how to use a microscope, and you know that if you take some living tissue or maybe some water from a pond and you put it underneath your microscope and view it, you're going to be able to see cells. So you know, and in fact, you know, up until this point, up until the late, you know, you've known since the 1600s or the late 1600s that living organisms were made out of cells. You know that there were cells in the body. And you said to yourself, all right, well, what I want to do is I want to study the cells of the nervous system. So I'm going to get some sample of nervous system tissue and I'm going to put it under a microscope. And you're expecting to see cells, but what you actually end up seeing is this. What you're going to find is that you're not able to easily identify really anything. You might be able to see some blood vessels or some other structures like that, but you're going to have a very difficult time seeing you know, where the cells are in your sample of nervous system tissue. And that was the state of things up until the late 1800s, when a scientist named Camillo Golgi discovered a stain that would selectively stain a small fraction of neurons and would stain them by filling their entire structure with this very sort of dark black silver nitrate substance. So if you get a hold of this Golgi staining method and you apply it to your nervous system tissue and then you put it underneath your microscope, finally you're going to get to see this. You're going to get to see what an actual neuron looks like. And I can't emphasize just how totally revolutionary this was. You know, again, since the 1600s, people had known that the body was made of cells, but it was still unclear up until the 1800s whether the brain, like the rest of the body, was made of cells and what those cells looked like and how they were structured. But with this stain, with this Golgi stain, finally, the structure of neurons was, you know, could be seen and could be studied and could be understood. And this sort of led to an explosion of new information about the brain and the nervous system, and basically led to every modern discovery we have about the brain. It led, you know, first of all, to our ability to be able to identify cells on the basis of their structure, and to identify different cell types on the basis of their structure. This is an illustration of Golgi-stained neurons in the retina, and we can see even in this one little tiny part of our nervous system contains this incredible diversity of neurons that all have these very, very distinct shapes, and as we would learn later on, distinct functions. In addition, this stain allowed people to begin for the first time to really see the organization of the nervous system. So this here is another illustration. I believe this one was by Golgi himself. This is a coronal section through a part of the brain called the hippocampus, which is deep in the temporal lobe. And what you can see here, if you look at how all of these different cells are organized, is that in different parts of this section here, there's a different level of organization. We can see that in this section here, there you know, maybe are two layers here that we can see. The organization isn't very clear. If we move on to this section here, very, very clear laminar organization, very clearly organized cells in this sort of neat row here. And then finally, if we move on around this curve, we see the organization breaks down. Maybe there's a layer here, but it's not as well organized. So, you know, without this, without being able to see how these cells are shaped and how they're organized, you'd have really no idea about the functional organization of this part of the brain. But now, with the Golgi stain, we have it. All right, that was just my little bit of neuroscience history. Thank you for indulging me. Let's get on to a picture of our neuron and talk about the structure of a neuron. Of course, a neuron, like every other cell, has a cell body, and the cell body contains the nucleus and the Golgi organ and the endoplasmic reticulum and all that important stuff that's important for making a cell do what it does. In addition to that, near the axon, we have this region that we call the axon hillock, or the axon initial segment. This area is important because the membrane around here contains a large concentration of those pumps and channels that I talked about. And as it turns out, this region is very, very important for initiating action potentials. So we'll learn about that later when we talk about the initiation of action potentials. Neurons, of course, have an axon, and the purpose of the axon is to carry the action potential away from the cell body towards the terminals. Axons can be incredibly long relative to the size of the cell body. Right, a cell body is going to be much smaller than a millimeter, whereas an axon, for example, the axon that innervates your your little toe, they are going to be going all the way from the bottom of your back down your leg out to your little toe, so they can be a meter or more in length. Axons can split, so one neuron can split its axon and therefore communicate with many different neurons. And axons tend to end in what are called either axon terminals or terminal buttons or terminal boutons. These are just little enlargements at the location of synapses that contain a lot of synaptic vesicles and other machinery for synaptic transmission, and we're going to be talking about that in a couple of lectures. Moving to the other end of the cell, we have the dendrites of the cell. We can see dendrites can form these large branching patterns. They can be very large in some cases, and in some cases they have what are called dendritic spines. You can sort of see them in this Golgi-stained neuron here. They're also illustrated here. Spines are like these little hairy projections coming off of the dendrites. And again, these are where synapses tend to form and contain a lot of the postsynaptic machinery in the case of the dendrites. All right, so this is sort of the basic structure. Almost all neurons have these basic components that I just talked about. Now, next we're going to talk about kinds of neurons, or different kinds of neurons. And really, we're not going to talk about too many different kinds of neurons. What I really want you to do is just understand the different ways that we can sort neurons or classify neurons. And in general, there are kind of two ways to do it. The first, and I mentioned this before, is looking at their structure or their appearance. And the reason is really simply that the structure can give us clues to how that neuron might function. So for example here, over here we have an example of a very distinctive looking neuron. This is called a Purkinje cell. These are cells that are in the cerebellum. And this is the axon of the Purkinje cell. And this is the Purkinje cell's dendritic tree. So you might imagine, simply by looking at this Purkinje cell, that it's going to be receiving input from a lot of neurons and be receiving a lot of synaptic input and integrating that input, and that's going to be a very important part of its function. And that would be absolutely right. Here is another kind of cell called a pyramidal cell. A pyramidal cell comes from the impression that some people get that the cell body looks a little bit like a pyramid, like a four-sided pyramid. And again, we see it has this dendritic tree, not nearly as large as the Purkinje cell. Over here we have a cell that's called a stellate cell. Stellate comes from the Latin for star, again because of its star-like shape. And it can be important, especially if you're looking at an area that hasn't been well studied before, to understand the structure of those neurons and how they're shaped. However, you also have to keep in mind that structure doesn't give you everything. And maybe another way to put it is, structure, it's not just a one-to-one relationship between structure and function. Neurons that have similar structures can do different things, or neurons that look very different can do similar things. So now for me, a better way, or maybe a more informative way of classifying neurons is based upon their function and upon their connectivity. So two very, I think, clear examples here are sensory neurons and motor neurons, right? So again, let's think of your little toe, right? There are pressure sensors and vibration sensors located in the skin of your little toe, and in order to sense those, detect those sensations, there needs to be a neuron that receives that information and transduces those sensory signals into action potentials. And so that is a sensory neuron, and we know what that neuron does because it is connected to a particular part of the body and, let's say, to a particular kind of sensory transducer. Likewise, motor neurons. These are neurons that live in your, usually in the ventral horn of your spinal cord gray matter, and send their axons out into your body and connect up to muscles or connect up to internal organs to either generate movement or to engage in some bodily functions. And we know from looking at the connectivity of this cell what it does. We know that it's connecting to muscles and that its cell body is in the spinal cord and that it therefore must have some motor function. And in fact, you might remember, we talked about sensory neurons and motor neurons when we talked about our basic reflex arc. Now the trick is, or maybe the difficult part comes in, to everything that's not a sensory neuron or a motor neuron. And in general, if it's not a sensory or motor neuron, we call it an interneuron. And what those neurons do is they're the ones that are connecting one neuron to another neuron, but themselves are not connected either to a motor end effector or are not connected to a sensory transducer. And the thing with interneurons is that really pretty much almost every neuron in the brain and in the spinal cord is an interneuron because it's not connecting to the sensory world and it's not sending out motor commands. And we can imagine that these interneurons form sort of really vast interconnected networks. And I want to sort of emphasize this. In this illustration here, this is our drawing of our neuron, and we see that it's getting some axonal input here, and it's also sending some output here to the dendrites of this neuron. And you might get the false impression that this is how the nervous system or the central nervous system is wired up, that we just have one neuron connecting to another in sort of a chain. And that's really not the case. Neurons are connected in very dense networks. And I just want to show you here an example of what I mean by that. So this is an illustration that I pulled out of a paper in a journal called PLOS Biology. This is, what we're looking at here is an illustration of a worm called C. elegans. C. elegans has a very simple nervous system. Contains, I think maybe something like 54 neurons or 254 neurons, not that many. And people study it because it's simple and because the connectivity of many of the neurons in the C. elegans nervous system are known. And what you see here is just a connectivity diagram for some neurons within C. elegans. So for example, we know that these are sensory neurons here because they receive input from touch receptors. And we know that these are motor neurons here because they connect to muscles, which means that these are interneurons. And we'll see here that it's not just a straight connection from sensory to interneuron to motor neuron. We see that the interneurons are connected to each other in complicated ways. By the way, you don't need to memorize this. You don't need to know anything about this diagram other than the fact that in real nervous systems, interneurons in the central nervous system, which is most of the neurons in the nervous system, form these complicated connected networks. And this is a challenge, right? Because, we know what a sensory neuron does because it's connected to our sensory transducers, right? So its connectivity tells us what it does. And we know what the motor neurons do because it's connected to muscles, right? So its connectivity tells us what it does. And the same thing is true for interneurons. Its role in the network is going to be in large part determined by its connectivity with other neurons. But because that connectivity can be so dense and so complex, it can be very hard to know what neurons do, the role that they serve within a network. And this is, in fact, one of the sort of great challenges in modern neuroscience, is trying to sort of pick apart the circuitry within the central nervous system and understand how that connectivity between neurons gives them their function and makes them do what they do, whatever role it is that they play, whether it's aiding with sensation or guiding movement or cognition or memory or something like that. So really, when you think of the central nervous system, when you think of the brain, and really this is any part of the brain, the cortex, the brainstem, even within the spinal cord, the neurons within them are connected to each other in networks, and those networks are dense and can have very complicated connectivity. All right. Now, there's just sort of one little point I want to make here. You may have heard of the term neural networks used in a different context, used in either computer science or applied mathematics. And you should just be aware that when you hear the term neural networks in a computer context, they don't mean biological networks. In computer science, artificial networks, neural networks are essentially artificial networks that are very loosely inspired by neurons and neural architecture. And for the most part, they're essentially used as a computational tool or as a way of studying artificial intelligence. Like a good example would be in your phone, probably has some voice recognition capabilities, and those voice recognition capabilities are usually going to be powered by a neural network, by an artificial neural network like this one. Okay, so now we've sort of talked in general, in very general terms, about the structure and function of neurons. We're going to finish up by talking about the glia. And maybe you've never heard of glia before, but glia are a very important part of nervous system function. So they have three main things that they do. First of all, they provide physical structure to the brain. And in fact, the word glia means glue. So in some sense, the glia have the role of gluing things into the brain together and holding neurons in place. They can help provide nutrition for neurons. They form what is called the blood-brain barrier, which we'll talk about later, and they are involved in healing and repair functions in the central nervous system. That's its sort of main function. Another important function in the nervous system is the production of myelin. And myelin, as we'll see when we talk about the action potential, is essentially a form of electrical insulation that helps neural transmission be more efficient. And finally, there's one particular kind of glia that secretes cerebrospinal fluid. And remember, cerebrospinal fluid is the fluid that fills the ventricles, and it actually surrounds the outside of the brain and provides buoyancy and cushioning to the brain. And one last sort of fun fact about glia is that if you just count up the number of glia and the number of neurons, they outnumber neurons 10 to 1. So you really have 10 times as many glia in your brain as you do neurons. I'm going to take a little aside here. You may have actually heard this sort of thing that, we only ever use 10% of our brain. I think maybe it might come from this idea that your brain is only 10% neurons, but in general, this is totally bogus and totally a myth. If you do any sort of functional scan of the brain that looks at brain function, what you find is that your whole brain is pretty much active all of the time, even when you're sleeping, and you're basically using all of your brain all of the time, rather than 10% of it. So anyway, you may have heard that myth and wondered where it came from. I wonder if it comes from this fact about the glia. I'm not sure. Anyway, just wanted to mention that real quickly. Now here are a few details about the function of glia, and I actually think this little chart in the textbook really, is a very nice, simple encapsulation of what glia do. So again, we have these glia that are secreting the cerebrospinal fluid. These are called the ependymal cells. The general support neurons are these neurons called astrocytes. They form the blood-brain barrier, which I'll talk about in a moment. Microglial cells are derived from blood tissue, and their general function is sort of cleaning up dead neural tissue as well as some repair functions. And finally, for creating myelin, we have what are called oligodendrocytes or oligodendroglial cells. These are the cells that form myelin in the central nervous system, and Schwann cells are the neurons that form myelin in the peripheral nervous system. So really, I think this little table here kind of encapsulates it all. One last thing I want to mention about the blood-brain barrier. The blood-brain barrier is an important feature which basically protects neurons and neural tissue from substances that are in the blood that could be harmful. And the idea of the blood-brain barrier, it's kind of hard to wrap your head around. It's not like a physical barrier, like a sheet of plastic wrap or something like that. What it describes is the fact that the capillaries within the brain have, of course, capillaries are formed by these endothelial cells. And in normal capillaries, in the rest of your body, these endothelial cells have a little bit of a gap between them. They're a little bit leaky, and this, of course, allows things and substances in your blood to get out and to supply your tissues. In the brain, however, the endothelial cells don't have leaky connections between them. They have, in fact, what are called tight junctions, and so these are protein junctions that link and tightly hold together the endothelial cells. And what that means is that there's less stuff that can leak out of the blood and get into the brain. And it means that neurons and neural tissue are going to be less accessible by things like antibiotics and drugs, but also less accessible by poisons and other harmful chemicals. This is illustrated here, sort of two ways of illustrating this. I actually like this one here a little bit better. So we can see here, if we take a cross-section through a capillary, there's blood on the inside. These are the endothelial cells that are sealed up with these tight junctions so that substances in the blood, if they want to get out, they have to diffuse through the endothelial cell. And when they do that, they can be taken up by the astrocytes, they can be taken up by neurons, or by these helper cells called pericytes. All right. You'll hear about the blood-brain barrier in medical contexts, and it's an important thing that the astrocytes help form. All right, so that brings us to the end. This is just our little summary and our learning objectives for today. So you should be very familiar with the basic properties of the membrane and have a familiarity with basic ionic chemistry. Again, these should have been in your prerequisites, so maybe consult some of that material if you find the textbook isn't adequate to get you up to speed. You should be able to understand the difference between a sensory neuron, a motor neuron, and an interneuron to be able to describe what makes them different. You should be able to name these basic parts of the neuron in your sleep. I'm not so worried about you being able to look at a neuron and identify what it is, but pyramidal neurons and Purkinje cells are probably two of the most iconic and recognizable kinds of neurons. So you should be able to pick those two out of a crowd, for example. And finally, you should understand the basic types of glia, the 5 types of glia and their basic function, and you should understand what the function of the blood-brain barrier is and how it's accomplished. All right, for the next lecture, we're actually going to be looking ahead a little bit. We're going to be looking a little bit into Chapter 4 to start laying the groundwork for understanding the neuron's most important function, which is the resting potential and action potential. Talk to you then. Bye-bye.